After you've permeabilized, then you go on to your blocking. And I'll spend a little bit of time talking here about the different blocking techniques. Usually when you say blocking, like for an antibody, you talk about protein blocking. If you use an antibody on tissue that's not blocked properly, that antibody can bind to proteins other than the antigen that you're targeting. And so instead, you first block with a protein blocker. In many cases, this can be bovine serum albumin or serum, like normal goat serum that's been heat inactivated. Sometimes out there, people will use others, like fish skin gelatin or non-fat dry milk, or commercial blockers like blockade. I have found that bovine serum albumin, maybe at a 6%, combined with maybe a 3 to 5% serum, is a very good combination to knock down some of that non-specific binding by the antibodies. However, here we sell a component called blockade, which you can purchase at a 1x concentration, and it's not species specific, unlike serum. So you can just wash your sample with that for 30 minutes or so to block, and then you can even use it in with your primary and secondary to block those as well. On the upper right, you can see some example images of different blocking conditions compared to each other. This is a cerebellum, mouse cerebellum section, a cryosection that's been labeled only with a secondary antibody with alexaflor 488. With BSA or NGS alone, you can see that you get a pretty high signal. In the graph there, those are the two left columns, BSA and then NGS, normal goat serum. If you use blockade, you see that you get even less nonspecific binding, and that's a good thing compared to autofluorescence. Protein blocking is very important in order to make sure that you get a specific, or as close to specific as possible, label. You don't want to confuse with the background. Now that's protein blocking. And remember that every assay is a little bit different. Every cell type is a little bit different in terms of the optimal blocking conditions. So I definitely recommend optimizing those on your end. Now, one type of blocking that people don't tend to realize that they need is due to dye charge. Most dyes have a negative charge on them. And because they are negatively charged, they're attracted to the positively charged areas on the cells or tissue. On cells, that's usually nuclei and mitochondria. On tissue, that can be, for instance, white matter in brain sections. There is a solution called the ImageFX signal enhancer that will block those positively charged areas, and therefore the negatively charged dyes won't bind to it. Here you can see some examples in this image at the top of with no blocking versus normal goat serum only, and then labeled only with a secondary antibody. And you can see, although it's a fairly long exposure time, you can see a fair amount of background there, and that's those two bars in the graph. But if you use the signal enhancer solution, you can see that it knocks it down pretty much to autofluorescence there. Very important, and something most people don't think about. Here also is some examples with that signal enhancer on tissue. I mentioned brain tissue. On the upper left in this image, you can see nonspecific binding by alexaflor 488 secondary on that highly charged white matter. But with the signal enhancer, it's knocked down to just about nothing. Very good. Remember, dye charge blocking means less nonspecific background. And then in addition to the protein blocking and dye charge blocking, there's some specialized techniques that require their own blocking steps as well. It's very common to use a streptavidin biotin amplification, and I'll talk more about amplification techniques in a minute. But if you use streptavidin biotin, you want to block the endogenous biotin in your cells or tissue first. Otherwise, you'll be labeling that as well, confusing your signal. In the image to the right here, you can see on the left some cells that have been blocked for endogenous biotin with one of our endogenous biotin blocking kits, and on the right, a cell that has not been. In the cell on the right, you can see you've got nonspecific binding to the endogenous biotin in the cells, mainly in the mitochondria, whereas the picture on the left that was blocked does not have that problem. And then also, if you use an enzymatic sort of detection, like with DAB or tyramide signal amplification, you want to block the endogenous enzymes in that mainly tissue. For instance, peroxidases. If you're using a horseradish peroxidase or HRP detection, like with diaminobenzidine on your tissue sections or with tyramides, you want to do a hydrogen peroxide wash to eradicate the endogenous peroxidases before you go through with your labeling. Phosphatases also, like for BCIP NBT detection, you want to reduce the endogenous phosphatases with levamisole. Always to get a specific detection.